then working in Toshiba in Japan. If you want to speak Japanese with him, he can. He's also reading only Japanese books. It's a little bit freaky, but still. And then he has been working in Switzerland and France in academia. He came back to Spain to work again in a company, in a small company, very research intensive, and now he's back at purely the academic side. So in that sense, he has a very interesting profile going from the different points of view to look at medical technology in general. He has been doing a lot of medical image analysis and has done a lot of basic work there, but he has also brought it further in order to look at therapy planning, for example. It's like, how can you use images and simulations, for example, to use the knowledge then in order to do therapy planning. And what he's going to talk about is a very kind of interesting example of this, where you try to model cochlear implants, which is in some ways a therapy that has to be planned and delivered, but on the other hand is also working in a very complex organ where both the shapes and the physiology are very important. So this gives a very, very nice overview of how you can integrate image analysis, computational modeling with knowledge of physiology in order to really deliver a therapy to the patient. Thank you very much for being here, and we're looking forward to your explanation.
Thanks very much for the introduction, Bart. So as Bart mentioned, I'm here since a few years ago in the group. And when I arrived at UPF in this department, which is a very multidisciplinary department, there was the first floor that was basically the biomedical engineering part. And if you go to the right of the stairs, you had the brain people, the Center for Brain and Cognition. And to the left, you had the cardiac people that were doing research in the heart. So it was clear from the beginning for me that I should be a brainless, heartless researcher here in the department. And the example I will show is research on the ear, which is a bit of a very exotic thing, even in this forum where we have seen research on the lungs, on the heart, on brain, etc. Incidentally, I do also research on brain and heart. But most of the work I will present is actually from the PhD work of Nerea Mangado, who was a PhD student in our group and is now a postdoctoral researcher. It's work also that was co-supervised by Gemma Piella, who is also co-director of our group. And also I would like to acknowledge the strong contribution of this towards this research that Mario Ceresa and other senior researchers in the group have done. As you see, the title is Image-Based Computational Modeling and Simulation of Cochlear Implants. So you see from the beginning that much of it will be about image processing. So my background, as you mentioned, is from image processing and computer vision. But then I will explain how to go about doing patient-specific computational models and how to do simulations of both the surgical insertion and also the functional activation of cochlear implants. Okay. So going towards, if I can, so this is the brainless part. I can't move. Okay. Is there any mouse or something I can use? There's something here. Maybe it should be connected somewhere. But I don't know if it will reach up. Okay, now it's moving. Thanks. Okay, so going back to your very basic elementary school biology, you'll remember that the hearing system is composed of the external part, so the outer ear, okay, where you have the visible part of the hearing, and where the sound waves actually make their way towards a membrane that is here, the tympanic membrane, towards the middle ear, which is this set of small bones that you will remember have funny names, like the stirrup and the hammer and these things. And basically they transduct the vibrations of this membrane to another membrane that is hitting on this organ in the inner ear. Oops, sorry. And the inner ear is composed of the cochlea, that is the part that captures the hearing, and the vestibular system, that is the one that takes care of balance, of the feeling of balance. Right? All this system is filled with liquid, okay? And the vibrations in this membrane actually are transmitted along this liquid, the paralymph. Okay. Then we will see how the cochlea actually makes these vibrations change into electrical signals that go towards the auditory nerve that actually make it to the brain, so you actually make sense of this hearing. Right, if you take the cochlea, which is this snail-shaped organ, and you make a cut through it, you will see that there are several cavities here. So actually there are 3 in each turn. Okay. All of them are filled with liquid, like we mentioned. There's the scala vestibuli, the scala tympani, and a medium part where the organ of Corti is. Okay. So as we see here, these pressure fluctuations that are transmitted through the vibration of sound towards the vibration of the liquid are the ones that are propagated inside the cochlea. And there are very funny things about the cochlea. And that's one take-home message I want you to take, that the ear is a very sexy structure to do research on. So one interesting fact is that the cochlea doesn't grow. So if you put an implant, whatever, for a baby or for a kid at a certain age, in other applications you have to think about people growing and maybe reimplantation and things like that. In cochlear implants, there are reasons for reimplantation, but they don't have to do with the actual growth of the organ. So when you're born, that's it. You don't grow. Your cochlea doesn't grow. Another very interesting fact is that the cochlea, the structure of the cochlea itself, has what is called tonotopical mapping. So the frequencies of the sounds are actually mapped along the spiral of the cochlea. So all the high-pitched sounds, like these, are captured at the beginning, right? And all the low-pitched sounds, like these, are captured at the tip of the cochlea. Okay? So this makes for a lot of interesting capabilities when you think about developing an implant, some sort of device, to be able to, stimulate the nerves that that actually are the ones that are transferring to the brain at the different locations along the spiral to actually create sound. Okay? Because it's quite direct. In a sense, it's a bit similar to vision as well. So if you go to the cortical part, so the visual cortex, you can kind of paint images on it. What else? As I said, within this there is the organ of Corti. So the organ of Corti is formed by ciliated cells that, when there are vibrations in a membrane that is here in the cochlea, these mechanical vibrations are translated into electrical stimulus. Yeah? And these electrical stimuli are the ones that go to the nerves and actually make for the signals that create the sound in the brain. What else? I think I jumped on the structure of this membrane. So this membrane that is located here actually has different properties along it. So as it goes from the start towards the tip, the vibrations that are induced have different properties, and this is what allows to actually capture the different frequencies at different locations. So this just to go towards the introduction of how the hearing system works. Okay? Hearing impairment, as you all know, is a serious condition. It has a huge impact, of course, at the personal level to people that suffer it. There is a high prevalence of babies that are born with certain conditions. So there is let's say the distribution of people that are implanted by cochlear implants is a bit like a U-shaped. So you have babies at small age, around the age of 2 or so is a bit the median age for surgeries. And then you have elder people that actually have conditions that lead to hearing loss, and then they are implanted at a later age. Still, there is a high prevalence, as you see here, over the age of 45 for higher age people. About 25% have some sort of hearing disability, and about 20% of them cannot be treated by conventional hearing aids that are basically microphones and speakers inserted in your ear, and have to go about doing a surgery and being implanted a cochlear implant. Okay. So how a cochlear implant works is by actually bypassing completely the hearing system chain. Okay. So some people say this is a bit of the first successful example of cyborg technology or bionic devices that actually replace completely human sense. Okay. Incidentally, companies that are working on these devices are also very active in research, working on things like visual implants, implants for replacing your vocal cords, so you recover your voice, and why not? You could think of implants to actually enhance your capabilities, like having superhero powers, like you mentioned. Okay. In any case, for the cochlear implants, what it has is it has a sternal part where the sound is captured with a microphone, where the sound is processed, and there is quite a lot of activity in terms of signal processing in this field, so trying to enhance how the audio is captured and then translated. This goes to an internal part of the implant that is implanted below the skin, and the information is passed to it, and then it goes through this electrode array, which is this flexible part that is inserted all the way inside the cochlea. I'm not sure if you can see from there, but actually this electrode array, it has small metallic parts along it. I'm not sure if you can see. So these are the ones that are activated. And you can see that depending on which electrode is activated, this corresponds to a location in the implant, and therefore it corresponds to a frequency of sound. So this is a bit how they play with stimulating the sound, with stimulating the nerves and generating the sound. Okay. So what else is there to do? It's a fairly successful device. It has been around for some years. There are a number of companies that are developing and selling and implanting these devices. However, it has been seen that there is quite a high degree of variability in people that are implanted. This depends on a number of factors, not least the actual morphology of the cochlea, the characteristics of the patients. So it should be interesting to explore possibilities for patient-specific planning for this sort of interventions. Okay. Other things that have to do with the optimization of the implantation is also not just the insertion itself, the surgical act, but also the activation plan. So the actual function on how to activate the different electrodes to best generate the sounds. Finally, I would like to mention also that there is a trend in novel approaches for surgery that try to insert the electrode to a certain depth only. Because people lose hearing at a certain age, starting from high frequency sounds. So usually older people, they start losing hearing, but still keep quite good hearing of low frequency sounds, of the bass. Yeah? So there are techniques of people that are able to actually insert the implant only at the beginning, which is the part of the cochlea that deals with the high frequency sounds, and while still keeping the cochlea active, while still keeping the liquid inside and allowing for vibrations. And of course, if you can keep natural hearing for part of the sound range, that has better qualities for the patient. So being able to plan these sort of insertions will also be interesting, because you would do it thinking how much you are going to insert the electrode. So I will go through a number of works we've done in this direction. I will basically explain two. I have a third one that if there is time, I will show a few ideas, but I'm not sure that time will allow it. So this is a paper that was published. As I said, Nerea Mangado was the first author of this paper. It was published in Annals of Biomedical Engineering. I don't have the impact factor. So automatic model generation framework for computational simulation of cochlear implants. Again, the motivation, I sort of already mentioned it, but the whole idea is to have an automatic framework for the generation of patient-specific finite element models to then be able to simulate how the implant is actually inserted, and then doing simulations, as we will see in the second work, of the function of the implant. You see the elements here. I will go through each of them in a bit more detail. The first element has to do with things that have been already commented by Blanca, also by Marin yesterday, on the use of statistical shape models as models of population variability. This is a line of research that, to me, has been a very extensive, let's say, line of work. So I've been working on this for a number of years. I think my first publication with statistical models is from '98 or so. So it's a model that I think has been particularly successful in capturing shape variability in a very, let's say, intuitive and practical way. So the way it works, well, in this particular work, we had a number of CT scans of patients. CT scans of patients are low-resolution scans, so you cannot see the details of the cochlea. So what we wanted to do was to do a statistical shape model with images that are higher resolution, which means that we got cadaveric samples of cochlea, of 17 cadavers in this case, and they were scanned in very high-resolution micro-CT devices. So you cannot do this with patients, because to do that you would have to cut the bone and put it inside the machine and then put it back in the patient, and that's not going to happen. So we did work together with Scanco, that is a company in Switzerland that was part of the European project within which we were doing this work, on actually optimizing the acquisition process, the image reconstruction, etc., worked on segmentation methods, and then on how to build the statistical shape model. In particular, for the segmentation work, we developed a number of tools. You can see here some of the references of papers that we created related to this work, because the type of data is a bit special. So we had data sets that were quite heavy, because micro-CT data generates volumes that are quite high. The level of detail is not uniform across all the structures, so you had to do some segmentation methods that actually take into account how much detail you want for each of the structures, because some of them, like the overall bone outline, has a quite soft shape, so you don't need very detailed segmentation there. But then to be able to segment the vascular membrane or the locations of the nerves, etc., you had to do quite special things. But in any case, to cut the story short, there were a couple of methodological approaches that we focused on. One was based on modified versions of level sets, and this was work of Martin Kier, who was working in DTU in Copenhagen, and also here. He did some stays with us. And also graph-based segmentation methods developed by Esmeralda Ruiz Pujadas, who was a postdoctoral researcher in our group too. So in the end, we have this beautiful cochlea that we can rotate in 3D and have all the detail and everything, but for the cadavers. So with this, as many of you are familiar, and you've heard this many times already, with this population of cochlea, we had to do registration, we had to align all the models to be able to then compute the average cochlea, kind of the typical shape of a cochlea. And then by analyzing the principal components of the covariance matrix, we had the modes of variation, so the variability around the cochlea. I have a small video here of the first mode of variation. Go the second mode of variation, etc. And showing this video, even though it's not actually the last version of the statistical model, showing this video has been quite interesting in clinical conferences. So just last week, I was in a clinical conference of people doing cochlear implantation, and a lot of people are working on characterizing the variability of the cochlea. And it's funny sometimes that work that is done, this was mentioned also by Blanca, work that is done in this community doesn't actually permeate to the clinical community. So they are doing statistics on the diameters, the maximum diameter, the smaller diameter, etc., and doing statistics on that, whereas there are engineering methods to study the variability of shape that are much more advanced and not necessarily difficult to manage. So anyway, with this statistical shape model, we can do two things. Well, we can do more things. We can generate funny videos, but the first mode of application of these statistical shape models is what I was mentioning. If you have a clinical CT where you see maybe, I'm not sure if you can see from there, that the structure of the cochlea is not very well visible, so it's very difficult to actually separate the different turns of the cochlea. So you can try to find what is the best instance of your statistical shape model that approximates the low resolution information you have in your clinical image. Okay? So you can see from these videos that we were doing that this is generating many different shapes. This is modulated by just a set of a few scalar values that actually are the ones that represent each of these shapes. So you do an optimization to find which are the values that generate the shape that best fits this low resolution data. Okay. So with this, we end up having a patient-specific model. The other use that we have from this statistical shape model is as a generator of cochleas, so a generator of virtual populations. And why is this interesting? Because we are working also together with implant manufacturers. And most of implant design processes nowadays, not only in cochlear implants, are based on tests on human cadavers, things like that, right? So being able to generate virtual populations allows them to test different implant designs, not only based on the geometry of the implant, but also, as we will see later, on the function, so the activation of the cochlear implant. Okay. So once we have our patient-specific representation, that is high resolution and everything, as I said, then we do the simulation of the implantation. We were mostly working with one of the manufacturers, but this can be used with any of the implant manufacturers. We had CAD models of their implant range, so they have different models of implant where they have different characteristics and also different locations of where the electrodes are within the electrode array. And by using just simple mechanical simulations, we were able to simulate the insertion inside the cochlea. Okay. There are a number of interesting things here to study just by focusing on this line. And this is something we are exploring now as future work, because there are theories about how to best insert the electrode array so you don't risk breaking the membrane. And this is one of the things that sometimes happens, and it really messes up the surgery. In any case, in our case, what we were doing were a first step by doing more, let's say, a heavier simulation of the insertion. In particular, we were using SOFA. And then we did a method to deform the CAD model. I will go a bit fast. So to modify the CAD model of the manufacturer in a way that it didn't introduce any geometrical distortions on the electrode array that would distort the activation of the simulation that we will do later. Right, I jumped this a bit fast for the sake of time. What else? So now we have the geometry of the cochlea, we have the different turns, we have the electrode inside, the structure of the cochlea. I'm not sure if you see here, I don't have a mouse or anything, but if you look at the top right there, you will see that there is this the yellow part that goes from the organ of Corti all the way down, that goes towards the brain. Okay, so this is the place where the nerves are, the nerve fibers. So what we were doing was also simulating the locations of the nerve fiber bundles, which are the ones that will allow us to simulate the action potentials, to simulate the activation of the nerves once the electrical simulations are performed. Okay? So to finish with this version of the work, this particular publication, what we did was to put this within a homogeneous sort of bone box to then give an environment where people could do simulations of the implantation. And there you have some details on the different properties we gave to the resistivity of the different materials. Okay, I will go fast. So these are some results. We tested on different types of implants, different types of insertion depths, the time that it takes to generate one of these models, which is fairly fast. And this was published and also left as open source for the community. So to move to the next work, which is the one I will want to focus a bit more now. One of the limitations of this work was, of course, the bone model. In the end, we just put a box there, so it's not really a patient. So we wanted to go beyond this, and also at the same time, we wanted, of course, to go towards the functional simulation of the implant, so to simulate the activation of the implant in terms of electrical activation and also nerve activation. So this is the second work. This is also work led by Nerea Mangado as first author, and this was published in Molecular Neurobiology last year. So it's towards a complete in silico assessment of the outcome of cochlear implantation surgery. Okay. As we mentioned, well, the motivation is clear. We want to do a computational framework to completely assess the patient-specific functional outcome of the surgery. So be able to plan the surgery, to simulate to create the model for the patient-specific and plan-specific situation, and then be able to simulate its function. Okay. So I will go through each of these steps: the computational model creation, the electrical simulations, and the auditory nerve fiber activation model. So for the computational model, we based this, of course, on the previous work. So all these first steps, the statistical shape model, the creation of the patient-specific cochlear shape, the virtual insertion, etc. That's pretty much what I just explained now. For the environment of the cochlea, what we did was a bit of a pragmatic solution, which is to take an existing model from the community. It's the VHP-CT dataset from the University of Iowa. And what we do is we insert our patient-specific model into this generalized model. Of course, it is not the shape of the head of the patient, but I think it's a fairly reasonable approximation to give realistic boundary conditions around the simulation of the implant. Right. Then for the electrical stimulation, I'm not going to go into much detail of things, but basically we did a simple approach where we were simulating the activation of the electrodes. There are a number of configurations that people do for electrode activation. So you can activate one of the electrodes and then use the reference electrode, which is this part that was close to the ear, as the ground. So you see here how the electric field is in that case. This is the most common type of activation that people do for cochlear implants. Then there are other types of activations where you use the ground actually in the neighboring electrodes. So you stimulate one and the other two ones are then used as ground. In this case, what I'm showing is a quasi-static approximation for the simulation, where you also have now a model for time-dependent simulations. What else? If we see here a cross-section of one of the simulations, so this would be this plane that I'm showing here. You can see the different isolines in the simulation. And what we do with this, of course, this gives an idea of how the electrical current is distributed in your head, okay, which is a bit, I don't know, scary or something. Of course, the part that we are interested in is how much of this activation gets to the different nerve fibers, okay, to then simulate how the activations of the neurons are done and how the information goes to the brain. Okay. For the nerve model, we didn't invent anything new. So we are based on existing simulation models. In this case, there was a previous work by Ratai et al. That already proposed a model for neurons within the context of cochlear implants. As you see, it is based on an analogy of an electrical circuit. So these are the type of simulations that we used. We modeled 47 compartments for each neuron. And we're also able somehow to tweak this model to then simulate auditory nerve fiber degeneration. And this is part of work that we did together with NASA Ames, close to San Francisco. They were interested in doing simulations with nerve degeneration for different applications. And we have a joint publication with them on the case of cochlear implants. Then for the activation itself, so of the effect of the action potential, and we basically use a standard Hodgkin-Huxley model to simulate the excitability of the neurons. Okay. So what do we do with this? As I said, the cochlea has this tonotopic mapping. Okay, so high frequencies at the beginning, remember, and low frequencies at the end. We can plot this in this type of visualization. So if we have here the number of electrodes from the beginning to the end, and we have here the frequencies, we can map the sort of ideal activation map. So the first electrode has low frequencies because it's the one that goes all the way to the end. And the last electrode, which is the one that is closer to the beginning of the cochlea, has an activation of high frequencies. This is the ideal thing, but then in real life, you don't have always this behavior. You can have this sort of pattern below, for example. And I think there is, sorry. So what we see in this pattern is that actually the spread of the excitation in terms of frequencies can be irregular. So you can have differences from the ideal behavior that have to do with the spread of the frequencies. You can see here an interesting effect, and is that when you want to excite low frequencies, maybe you are also exciting high frequencies. And this has to do with the shape of the cochlea. Remember, it's like a snail, okay? So when you try to activate, I'm sorry, the pointer is not very good at this, but if you look at the picture there, top right, you can see that different turns of the cochlea are sometimes quite close. So the nerve fibers go through the center, okay? And they have to reach both the bottom part of the cochlea and the top part of the cochlea. So sometimes when you're trying to excite a location at the, let's say, the tip of the cochlea, maybe the spread of the potential is actually also activating neighboring neurons that have to that correspond to lower locations of the cochlea. So this is what is called crosstalk effect. So you're activating, you're trying to reproduce a particular type of sound, but actually what you're doing is exciting at the same time high frequencies or low frequencies. So this leads to sound confusion in the patient. So we can, of course, simulate also this type of behaviors. We derived a set of measures just to see how to quantify a bit how close or how far we are from the ideal case. Okay. Other things that this method allows us to do is a bit of a shootout between the two main types of implants that are in the market. So there is the perimodular type, which is the one in the bottom, that is trying to force the implant to get as close as possible to the internal wall of the cochlea. So these are kind of pre-shaped implants. It's a bit like a coil. So when you insert it, it sticks towards the inside of the cochlea. And the theory is that because this is the part that is closer to the neurons, to the nerves, then it's the part that is most important. So the activation will be more selective. Other manufacturers say that this doesn't really matter, and on top of that, the mechanical stress that this puts in the cochlea actually has an effect that is bad for the patient. So with this type of framework, we're able to simulate both approaches and then simulate what is in fact the difference in activation that is gained by having these perimodular activities. So this, I'm not going to give details about the results. In fact, some of it is confidential because they wanted to have sort of munition to fight each other, let's say. Okay, I will jump this again, so I don't want to get too late for lunch. 1:30. Huh? What we did in these two works is, again, to have a complete framework for the patient-specific surgical planning and also functional assessment of the cochlear implant. We proposed some computational measures of goodness of fit, let's say, that allow to quantify how close or how far we are from the ideal implantation scenario, including these activation maps that we showed briefly. In terms of validation, which is something I didn't quite comment here, so this is part of it has already been done within the European project, and part of it is still ongoing. So we have measures from the patients in terms of the evoked response that patients have to the activations once they are implanted, to see if this correlates with the simulated activations. We are developing also some sort of phantom type simulations for the overall electrical activity, electrical activation to compare. And in terms of the mechanical insertion, so we did some validation by having samples scanned both in micro-CT and in CT pre and post implantation to see how realistic the planning was. And also this has been coupled with work of people that were also part of the European project that developed a surgical robot to do direct cochlear access. So basically with this type of framework allows you to have detailed plans for the surgery to see where really you want to insert and how deep. So this can be coupled also with robots from the University of Bern that allow to do minimal invasive surgery. So basically a drill that goes all the way to the part of the cochlea that you want to access instead of opening a big hole, which is what is done nowadays. Right, the third part, which I said I would mention very quickly, and I will, has to do with a bit of a mania that I have also from many years, which is studying the effect of uncertainty and somehow also variability. So we know that when we do a simulation, we can put some values to all the parameters and then get a response. Okay. But the information you have to generate the simulations has a certain origin. So you have maybe parameters from the literature that have been gathered by experimental means, where usually, if the work is good, you have a mean and a standard deviation for each of the parameters. Imaging data in itself, by the way it's acquired, it also has a certain uncertainty. You have voxel size, you have parameters that have to do with the reconstruction algorithms that finally generate the image. And of course, the reconstruction in itself, for example, here in this case, we are generating a high resolution model from a low resolution image. So that in itself has to have a range of variability that you can more or less approximate or quantify. So this part where in the particular case of the cochlear implants is a publication also by Nerea in Frontiers in Physiology last year. And she also wrote a review, which maybe is interesting as background if you're interested in these topics, on methods for uncertainty and variability modeling and propagation within finite element and computational simulations for different applications. Very quick, what type of variability are we talking about? So we have, on one side, we have uncertainty, that is information that we are not sure about, but maybe we can quantify a range. So we know that the parameter of the resistivity of a certain tissue is such, but we know that it has to be within a certain range. And also we can have variability. So we can have, for example, we can want to do simulations on a particular setup, taking into account that there is a certain variability in the population and see how this propagates in the simulations. Again, I go fast. In the particular case of cochlear implants, so we have variability in cochlear morphology, of course, of the population. We can have uncertainty and variability, both, in terms of the surgical plan. So variability in the sense that people have different approaches regarding how much depth of insertion they do, but also uncertainty, because once you have decided this insertion depth, you also have a certain play, let's say, on how accurate this is once you go to the operating room. And then other parameters such as the bone electrical properties or the properties of tissues in general. So very fast, what we did is take into account, for example, for the cochlear morphology, we can play on the parameters on the statistical shape model. So instead of giving a particular value, we can maybe define a range and explore around this range of parameters as a sort of neighborhood of the shape around the shape that we hypothesized. For the insertion length also, our framework allows naturally to simulate at different lengths. So if we design something like a Gaussian distribution of the parameter for the insertion depth, then we can simulate around it. And the same for the bone electrical resistivity. This is part of the state of the art, so I refer to the literature review for it. Basically, we studied two main approaches, which are the main two approaches that are a bit in the community. One is getting really deep into the maths and doing intrusive methods. So actually working with the equations to convert all the equations of the simulation to stochastic equations. This is what is called intrusive methods, and very hardcore mathematics people can do it. I can't. So we fought with it a few times. And it's still in the plan, but a lot of people told us, no, go to non-intrusive and you'll be safer. Non-intrusive has to do with sampling around a certain neighborhood. Of course, let's say the dummy way is by Monte Carlo. You can sample many times. You have the sort of smart Monte Carlos that are a bit more adaptive. And one thing that we did is collaborating with people here in Barcelona, in UPC, in CIMNE, which is a center for computational methods in engineering, tested some of the approaches that they have proposed that are called probabilistic collocation. And it's a bit like a response surface method. It's something that is trying to fit a function to the different outputs that you have by sampling the different parameters. So each instance of these parametrizations that you are exploring generates a simulation. So you can generate many simulations and then fit a surface. But their method is a bit more intelligent because it's an adaptive method that tells you how many samples actually you need to generate and where to best approximate this surface up to a certain accuracy. And what you end up having is a mean and a standard deviation for each of the parameters that you are simulating, let's say, or the outputs parameters. So we did this with our simulations. Again, I will just jump, sorry, but I think because of time I will not get into explaining this. Just saying, for example, we did a bit of a shootout also with Monte Carlo and this method. And we saw, for example, that one took 1,100 hours of simulation, and the other 36 hours of simulation with sort of comparable accuracy. And this allows also to do some conclusions, let's say, around what is the best way to tune the insertion and the activation of the implants. So to conclude, with this, we showed some work on how to incorporate uncertainty and parameter variability into this sort of simulations. And as global conclusions, what I showed was first a framework based on statistical shape models and also, let's say, classical simulations and model generation methods on how to do a patient-specific model for simulation of cochlear implantation. Then the second part was this part that refined a bit the model to make it more realistic, but also incorporated the electrical simulation, the simulation of the nerve fiber activations, and also the quantification a bit of the output measures, let's say, post-implantation. And finally, although I didn't quite explain it in detail, that there was this idea of how to incorporate, or the idea that work needs to be done to incorporate uncertainty, and also maybe within the same framework, methods have to be done to explore parameter variability for the particular case of cochlear implantation evaluation. Finally, just to mention also, we are doing some work towards tech transfer of this. So we did this together with clinicians, of course, and we are continuing on this collaborations with different clinics towards doing a software that is usable by them and can be used for patient planning, but also for more, let's say, fundamental research on cochlear implantation. We found that this is not totally incompatible with open science, and in fact, a big chunk of the research of the results that I showed here have been left open in a publication in Nature Scientific Data, which is a journal. Well, the journal is called Scientific Data. It's from the Nature group, where you can publish your models, your research. Leave it open. Each instance has a DOI to access it, and on top of that is a publication with a very good impact factor. So, and just to mention also, I already did, but a big part of this work that we presented was part of a European project that was called Hear You, led by us in collaboration with the company Alma here in Barcelona, Medel, which is the implant manufacturer I was talking about. They are from Austria, and they are the world's #2 implant manufacturer. The other one is from Australia, so all countries starting with AU are the leaders. Scanco, which is the company that makes the micro-CT devices. And as I mentioned, University of Bern, both the clinical side and the technical side, so the part that was doing the robot, and DTU in Copenhagen, collaborating with us on the image analysis part. And that's it. I would like to thank you. Yeah.
Thank you very much for this comprehensive overview of a nice integrated application. Are there any questions?
This is amazing. So I just want to know, so you clearly are having clinical impact, and my question is whether you have been able to quantify it. So one of the ways we can show significance of the work, if we are to get rid of impact factor, for example, is to be able to show impact, like you have. But we are asked to quantify it. Have you done any work on that?
I would say no. At the same time, maybe this gives the impression that we are in routine clinical use, which we are not. Of course, we have some pilot clinicians that are working with us, and we hope eventually we'll get. Also, the work has like different stages, and this is something that I always like to think when we have a European project proposal or something like that. There are results that can be applied fairly soon, right? So for example, the more geometrical part, the simulation of insertion, planning, and things like that, that we did some quantification with clinical cases in Bern. And you can actually compare with post-implantation images and things like that to see how close or how far you were. Of course, because this is kind of prototype technology, we were not able to quantify things like time savings and all that, which of course is an indicator that is always very valued by the direction of hospitals. For the more simulation part, this is more preliminary in the sense that we don't have really cases that we can quantify yet. It's part of the plan. But.
So just to follow up on that, so one of the things that I'm really considering is not only clinical impact, but the impact you have on the patient. And I do wonder whether you are talking to patient organizations. So because it's not just about how long the procedure takes, but it's also the impact it has on the patient quality of life. And I assume that having a big hole rather than a small hole has in your head has big implications in terms of
Yeah,
patient
in fact,
experience?
in the conference last week, we had like different populations. Yeah, there were the surgeons, the otologists also that do the actual fitting, because the thing with cochlear implants is that there is an adaptation period, because suddenly these people hear, and the brain plasticity and the whole thing is not a static entity. During 6 months or so, people change a lot. And then, of course, the patients. So with the patients, we had some initial contacts with patient associations. It's true that it's something that we want to promote more. So Jerome and I actually are leading a chair here in UPF that is funded by a foundation that is very interested in promoting these sort of activities together with patient associations. And during the conference, there was a patient association there with a small table in the conference. And I agree with you. I think it's something that more and more we should consider. At the same time, it's not evident how to involve them. So involving them, for example, in a European project, I'm not sure how, some of them are very receptive. At the same time, it can generate a bit of expectations that are difficult to meet. I don't know if you have experience on...
I mean, I'm just starting to think about it, but we do have patient and public involvement representatives in the more clinical research.
Okay.
And I think it's a really important thing to have now in most of the projects.
I think...
I want to add something to that. So in the Netherlands, we have a lot of involvement of patient participations. So the way funding works is a little different in the Netherlands as well. So we actually go door to door with collecting money from people, from the general public, for different causes. And so I work with the Dutch Arthritis Foundation, which is one of the causes. And so we collect money, and also we get a lot of money from donations, people that die. And so starting last year, there's two new things that Dutch Arthritis Foundation does. One is it's actually financially contributing to European projects. And the other thing is that we now, for every grant discussion that we have, there is a panel of patients present that are representatives of their patient group. And because within the rheumatic world, there's a lot of different diseases, like they actually read all the grant proposals. They specifically look for what is the patient participation. And I perform, like many of us, very fundamental research, and I found it really hard to see how patients could help me. They really positively surprised me. They were very involved. They knew much more about the impact of certain therapies, or even at the molecular level, than many of us in that room, because we're not experts on their disease. But many of these patient organizations are willing to contribute financially and with man-hour to the studies, and not as subjects so much, but actually people saying, okay, you're asking for donor material and you think it's not a big impact, but it's a huge impact. Or you're asking for me to undergo an MRI scan every so many months. But this, impacts me greatly. And it's all things that we hadn't considered. And I think it's really good to go to these, talk to the patient organizations once in a while and see how they could contribute to your work.
Thank you. I agree. I think it would be very interesting for all of us, and not least for the PhD students that get also this exposure and motivates them towards going towards having value in the final application. So I think it's...
Yeah, so a little bit adding on that. Well, actually, recently, so in the framework that Miguel Ángel mentioned, in this chair, so we've been in touch. So with the foundation, that is in direct touch with a patient association, and in arthritis. So in arthritis, there is not much very applied thing we can do directly to the patient. It's quickly becoming overcomplicated, and variability, etc. However, in very basic research, so this foundation was really insisting that patient association, so very much to discuss what happens at a very basic level. So it's not, they're not thinking about them directly, but they're thinking about how then the condition they're affected by might evolve in the next century, in terms of improved management. So here again, then the take-home message is to never think that research you're doing is too fundamental so that patients can be interested, actually.
Is this working? Yeah. I have another question about uncertainty quantification, because it's a new trend for all of us, and we become really interested in the methodologies. But I still need to have an example of where this uncertainty quantification has made a big impact. So methodologically is very interesting. We are all really interested. But the big applications I've seen of modeling and simulation that actually goes to the clinic have oversimplified a lot of the parameter complexity. And so what do we do with that uncertainty quantification? So imagine that we have solved the problem. You can do stochastic collocation. You can do everything you want. So what?
Right, yeah. I always say that in the end, the way you quantify how successful your simulation is or whatever has to do also with what you do with it, right? So, for example, for surgical planning, we tend to go towards, we want an accuracy of 0.000 whatever, right? But then the surgeon comes and says, well, actually the range of accuracy I have when doing the intervention is 1 centimeter, so, and this is so I see, for example, for surgical planning, for the insertion of the electrode, it would be good to actually quantify what is your accuracy when inserting it and what effect that has actually on hearing. So I do see your point. Yeah? At the same time, I think it's an interesting line. So beyond the methodological, divertimenti of maths. Yeah?
Hi, I have a question also about uncertainty quantification, because you distinguished in your talk between the natural variability and uncertainty, but then your methods distinguish between these two sources, or not really? Because I saw that you have just like one variable that is, for example, due to the shape, and it doesn't matter if it's due to natural variability and the error in the segmentation, or...
Right, it's a bit of a philosophical issue, I agree, yes, because it can be something that if you want to explore the variability in the population, then it's variability. If you are focused on a particular patient-specific planning, it's uncertainty, whereas it's still the same variable. So I agree with your point. There are sources of uncertainty that have to do, for example, with the imaging characteristics of the image of the patient, etc., where, of course, it's uncertainty and not variability. But in this type of variables, yeah, I agree with your point. A lot of things are philosophical when you get into this, and I really like your point about working with philosophy of science type of people, because I have the feeling that we are reinventing the wheel constantly. So all this new trend of machine learning, deep learning, etc., it goes. We've been, millennia going through the loop of models versus empiricist approaches, to then go back to models, Platonic things, to then. So I think it would be good to do an exploratory work with these profiles, as you mentioned.
Yeah, maybe as a last comment on uncertainty, for Anish. So I think that, so for example, so here this is an example of very applied case, no, where you would like to use the simulations in order to optimize a certain practice, and there are many factors you can play on in order to optimize it. And so here, it's I think a great application, and Miguel, you can correct me if I'm wrong, is then to use the propagation of uncertainty actually in order to mathematically and quantitatively then spot the few factors that would be adjusted, so and leading to specific recommendation to the medical doctor.
Thank you very much. I think we can finish. Maybe just one last comment on this is like, keep in mind that whenever you work with clinical data, the value that you get is wrong plus minus 5% of that value. This is a general rule. It works for lab results, for imaging results, for measurements of blood pressure, whatever. That seems to be a golden rule, and keep that in mind. It's very important for your understanding. Okay, I think it's lunchtime now.
Yep.
So when do we come back, Jerome?